first grand rounds of 2021. And once again, continuing our mission, doing this, whether it be in person or by Zoom, we're sticking to the schedule and making sure that we advance the mission and of disseminating knowledge through this forum. Today is a really special occasion because it represents something I think we all look forward to, the annual Frisbee Lectureship. And actually, to begin the forum, I'm going to turn it over to Ed Schneider. Dr. Schneider, as you know, is a professor of laboratory medicine, Cancer Center Director for membership, Director of membership for the Cancer Center, a leader of blood banking, certainly has done a lot of work over the years in that domain and has really been the shepherd for this lectureship for the past 18 years. And I want to turn it over to Ed to share some perspectives and as well introduce members of the family.
Thank you very much, Charlie. It's a pleasure to introduce the Frisbees for this lectureship. Rick and Chris Frisbie, son, Richie, developed leukemia as a young man, as a teenager, and was the first bone marrow transplant done in Connecticut. It was done by the late Joel Rappaport, who was running the program at the time. Richie did not survive. His first transplant failed and didn't survive to have a second transplant, which was to be his sister. And in honor of him and in his memory, the family, Rick and Christine, set up the Frisbie Foundation in 1990. They've given millions of dollars in cancer research and cancer education. They established the first stem cell processing lab at Yale, New Haven, long before Smilo had the first brick laid on the foundation. And that was the precursor of the ACT lab, which is currently supporting Smilo through the Department of Laboratory Medicine, run by Dr. Diane Krause. This lectureship was established 18 years ago, and the current speaker, Dr. Markus Muschen, is therefore the 18th speaker. And we welcome him, and we would like to turn it over to Christine to say a few words.
I'll be brief. I just wanted to really thank Yale, Dr. Muschen, and in particular Ed Schneider, who's been very close to us for many, many years and been very supportive of the foundation and the work we do. And we love this lectureship. The foundation's much smaller now. We don't do that many things, but this is one thing that we continue doing. And we're going to continue to fund this for years to come because we think it's just very rewarding. So thank you very much, Ed, Dr. Fox, and Marcus as well, Dr. Muschen.
Well, thank you, Rick and Chris, for all that you've done and for so much of the work that is being done for the patients at Smilo. We can thank you for setting the foundation for this. So I'll now turn it over to Dr. Fuchs to introduce our 18th Frisbee lecturer, Dr. Markus Muschen.
Ed, thank you. And Rick and Christine, thank you for your continued support of this important lectureship over the past 18 years. I think this has really been a wonderful tradition because what this lectureship has done is brought to Yale really a rich cadre of innovators in developing an understanding and new approaches to hematologic malignancies, which I think is a fitting legacy. And this year's annual Frisbee lecture is no exception to that impressive and august list of lecturers. Dr. Markus Muschen was previously the chair of the Department of Systems Biology and the lead professor at the City of Hope Cancer Center, as well as the Associate Director of Basic Science. And we were very privileged in the fall of 2020 to bring and recruit Dr. Muschen to Yale as our inaugural director for the Center of Molecular and Cellular Oncology at the Yale Cancer Center and Smilo Cancer Hospital, as well as the author and Isabel Bunker Professor of Medicine focused in hematology. Dr. Muschen trained in hematology oncology. His work, his training was both in the biology of hematologic malignancies and immunobiology. And frankly, over the past decade or really longer, he has been a leading innovator in understanding the evolution of B-cell malignancies, understanding the biology in terms of delivering new approaches to drug discovery in these cancers, and really now advancing that beyond in terms of the immunobiology and immunotherapy that would be available for lymphomas and hematologic malignancies, most notably now even in terms of novel CAR T therapies. And I think all of that really speaks to, in many respects, what's so relevant for the Frisbee lectureship. Markus, for his many accomplishments and an impressive publication record, has received countless awards. I wouldn't want to take too much time enunciating all of them, but they include the NCI Outstanding Investigator Award, a Howard Hughes Scholar Award, a Leukemia Lymphoma Society Scholar Award, Wellcome Trust Scholar Award, among many other awards that recognize his accomplishment and and record of innovation and accomplishment across all of these cancers. So it's really a pleasure to introduce Markus and in many respects welcome him to the faculty of Yale Cancer Center. Markus, thank you.
Thank you. I'm now sharing my screen. Great. Okay. Does this work? Okay. So first of all, I would like to thank the Frisbee family and Charlie and Ed for me being here. And since I came to Yale Cancer Center last fall, I came to experience that many of us who are devoted to the cause of childhood leukemia and leukemia in young adults, that the legacy of Richard D. Frisbie has inspired many of us. And today I would like to take the opportunity to present a few new findings from our lab that over the past recent years have led to a new concept that I hope will help us in the future to treat these diseases more efficiently than we were able to do in the past. And many of childhood leukemias are actually derived from B lymphocytes. And B-cell leukemia, in fact, represents the most frequent type of cancer in children and young adults. And one potential reason for that is that B cells during the early development have to go through a series of genetic modifications, DNA recombination, class switching, hypermutation, with the end goal for these cells to protect us by generating high affinity antibodies. And this is a cartoon here from 1905 drawn by Paul Ehrlich, who's sitting here in his office. And for all those reasons, B cells are in an extremely high risk for malignant transformation, actually 500 times higher than any other somatic cell. And for this reason, and also because humans can actually live without B lymphocytes for quite some time, we developed a research program that is centered on specific vulnerabilities of this very cell type. So as cancer researchers, we are always looking for vulnerabilities. And in this case, we are looking for vulnerabilities that are intrinsically encoded in the nature of B cells making antibodies and are selected. And B cell selection is the theme that I hope I will be able to present to you on the occasion of this lecture. The reason is that just by the random nature of recombination events of antibody encoding molecules, the vast majority, about 75% of all newly generated B cells are initially autoreactive, meaning that they're directed against self, and these cells have to be removed from the repertoire. And this means there have to be powerful mechanisms in place during normal development to destroy and delete these cells from the repertoire. And the overarching theme for our research in recent years is, can we actually leverage these mechanisms that are inbuilt in the life and selection and development of normal B lymphocytes for the treatment of B-cell leukemia and lymphoma. So in fact, the principle of B-cell selection is driven by signals from the B-cell receptor or surface immunoglobulin. And we like to think of this like as a Goldilocks principle, because only if the signal strength that is elicited from this unit here is just right, then these cells receive a positive signal and proliferate and survive. And this is usually the case when we have a balance between activation signals, namely kinases and phosphatases, that achieve this balance. So if the signal is too weak, for instance, if the phosphatases are too strong or the receptor itself is not functional, then the cells die by neglect. Now we are focusing here on the other end of the scale where the signal is overwhelmingly strong, which is typically the case when this receptor here is engaged by self-antigen, meaning that these receptors are autoreactive, and these cells could give rise to autoimmune disease and have to be eliminated. And so this principle is not only relevant to normal B-cell development. This cartoon here from a recent publication shows that in transformed B cells in leukemia and lymphoma, this signaling pathway downstream of the B cell receptor is engaged by mutations every step of the way. And so today I would like to divide my talk in 3 areas where we gain information of how we can leverage selection for therapeutic benefits. One comes from insight that we glean from mutations and deletions in B cell tumors, such as leukemia and lymphoma. Then insights from clinical trials. So we collaborate with large clinical trial groups in the United States and internationally, and look for predictors of clinical outcomes and what we can learn in terms of therapeutic targeting options. And then finally, how these complicated oncogenic signaling pathways intersect and how we can leverage these interactions again to undermine oncogenic signaling in these diseases. So in the first part, I'm going to talk about genetic lesions and what we have done here based on mutation data from COSMIC and other sources, assembled a set of more than 5 million somatic mutations in 39 different types of cancer, and look at these mutations from the angle of whether the mutation introduce a replacement or affect the coding capacity of the gene, or whether they are silent, meaning they are not selected for. And then in all these diseases, we rank the mutations based on these replacement over silent ratios. And when we do that in a way that ranks this based on B-cell specific ratios, we end up with typically mutated genes, and these are widely known in B-cell tumors like MYD88, A20, and so forth. But what's interesting to us that here at the top of the list, there are actually molecules in the PI3 kinase pathway that are frequently mutated throughout cancer, but are unexpectedly spared or exempted from in B-cell tumors. And these are activating mutations of the PI3 kinase pathway. And P10 and SHIP1 are inhibitory phosphatases in this pathway, and this is the catalytic subunit of PI3 kinase itself. So we studied this in multiple different directions, but I would like to focus here on P10 because it was just such a striking example. And as we know for a long time, P10 deletions and mutations widely occur throughout many cancer types. But in 925 cases of B-cell lineage leukemia, we didn't find any of these mutations, again highlighting the specificity of this mechanism. And P10 opposes PI3 kinase signaling by targeting PIP3, which is the central initiator of the PI3 kinase signaling pathway, and I will come to that back later in my talk. And to study hyperactivation of the PI3 kinase pathway by deletion of P10, we developed a mouse model in our lab based on conditional deletion of the P10 gene in our leukemia model. And unlike what we know in solid tumors, conditional deletion of P10 results in rapid cell death of those leukemia cells. And if we change the sequence and first delete P10 in normal B cells and then bring in a transforming oncogene, then we basically compromise malignant transformation. And then most importantly, when we wait for leukemia to establish in mice that bear these tumors and then delete the P10 gene, this leads to remission and the mice survive for indefinite periods of time. We also confirm that the biochemistry or the premise here is correct because we actually do see increased activity, increased output of the PI3 kinase pathway by increased phosphorylation of AKT. And most importantly, we used inhibitors that block the PI3 kinase signaling pathways at multiple levels here. Activation of PI3 kinase by SYK using the SYK kinase inhibitor and lusplatinib. BKM120 is a pan-PI3 kinase inhibitor, and AZD5363 inhibits AKT, and all three of them have in common that they actually rescue or protect leukemia cells from cell death that would otherwise be induced upon deletion of the P10 phosphatase. So this doesn't mean that these compounds are counterproductive in leukemia, because they're actually quite useful. Our interpretation of this unexpected result is that the introduction of those inhibitors restores signaling equilibrium, again, the Goldilocks principle that these cells need, whereas deletion of P10 alone here introduces a drastic perturbation, which engages negative selection just as it does for the elimination of autoreactive B cells. We are hopeful that these ideas, these concepts will eventually make their way into the clinic, and as an early indication that that might indeed be the case, I'm showing you two promising preclinical results in our lab, both based on small molecule inhibitors of these key phosphatases, namely SHIP1, inhibited by 3-alpha aminocholestane, and then P10, which also is targetable with a small molecule inhibitor, and both of them have desirable on-target activity biochemically, and importantly, they achieve disease control or disease burden control for long periods of time, and achieves extension or prolongation of overall survival of mice that bear patient-derived xenografts from B-cell lineage leukemia cells. So we are hoping that this approach can be developed further, and that some of these compounds will make it into the clinical arena. Now, the central premise of this idea is that this is a mechanism, negative selection, removal of autoreactive cells, that is uniquely important in B lymphocytes. And so to test this premise, we performed a reprogramming experiment in which we introduced CVP alpha, which is a transcription factor that can transform B cells into macrophages. And this is shown here. B cells express CD19, and this marker here is lost over time after induction and in favor of MAC1, which is a macrophage marker. And indeed, after some time, these cells start to crawl around on the bottom of these dishes and like macrophages, and can even phagocytose. And importantly, coming back to our hypothesis, if we genetically delete P10, even though genetically identical, the reprogramming from B to myeloid or B to macrophage fate almost entirely removes the sensitivity of these cells to removal of those phosphatases, which makes sense because macrophages don't make autoantibodies. There's no need for macrophages to be negatively selected as B cells are. So this gives us confidence that this is a real mechanism that is reflective of the nature of the immune system to purge autoreactive cells, and that is possible to selectively target this vulnerability in B cell tumors. Now, this work was done by Zhenjiang Chen in our lab, and he worried that this is not just counterintuitive, because you're essentially doing the opposite from what everyone else is doing in the field, namely by instead of inhibiting kinases, we are hyperactivating kinases. But most importantly, what was worried about what happens if we hyperactivate kinases for long periods of time, because that in itself could be dangerous. So he did an experiment to figure out what is the shortest period of time to commit B cells to cell death. And he did this with an engineered hyperactive form of the Syk kinase, labeled here with GFP. If you bring in this hyperactive kinase in the presence of a Syk kinase inhibitor, the GFP labeled cells remain constant because there's no hyperactivation of the pathway. Now, if we wash out the inhibitor, the cells, as expected, rapidly die. And here you found that if there's a lapse of just 3 hours, so removal of the inhibitor for 3 hours and then adding it right back, that already is sufficient to commit the cells. So that will be our goal going forward, to target a short, strong exposure to hyperactivation, probably with glucuronidated compounds that have a short plasma half-life. Now, in the second part of my talk, I'm going to give you 2 examples of how we can learn from information within clinical trials and gene expression annotation related to outcome. So our approach is based on microarray data that we obtain in collaboration with clinical study groups, and much of this work is also publicly available. And for each of these microarray probe sets that measure expression of individual transcripts, we divide the patient cohorts into 2 groups based on higher than median versus lower than median expression in these clinical trials. And then we ask the question, is there a difference between those 2 groups in terms of clinical outcome? And if the outcome is more favorable, we have here a blue annotation, and if the outcome is more poor, shorter overall survival, for instance, we have a red annotation. And if we group this heat map based on B-cell specific annotations, we come up with a list of genes that became interesting to us. And here at the very top is one molecule that I'm going to spend the next couple minutes on, the IL-2 receptor alpha chain, also known as CD25. So that was a bit unexpected, because CD25 is known as one of the 3 chains of the IL-2 receptor, and it typically pairs with the beta chain and the gamma chain to form a trimeric receptor. And this receptor is active on T cells and NK cells, and it's also important for formation of regulatory T cells, or Tregs, and therefore important to prevent autoimmunity, again, important to B-cell selection. And here I'm showing you that if you look at individual B-cell diseases, pediatric B-cell leukemia, CLL, adult B-cell leukemia, mantle cell lymphoma, we see a consistent pattern that the lower half of expression is related to better outcome, the top half towards outcome. The other reason we became interested is that if we bring in oncogenes that drive leukemia and lymphoma, like BCR-ABL or LMP2A, this leads to upregulation of CD25 on the cell surface. And also it seems to play a role in B-cell development itself. So if we profile here CD25 mRNA levels over the course of B-cell development, we find here a striking peak at a so-called fraction D. And that's interesting, because if you look at CD25 knockout mice, which we did in our lab, and compare this to the wild type animals and look at these fractions, we see here is fraction D, but this fraction is entirely missing in the knockout mice. And you can also see this here. This is a defect here. We still don't know what this actually means, but we also find that later in development, these cells actually are overrepresented in fraction F. So our initial hypothesis was this might reflect previously unrecognized role of IL-2 signaling in B cells. And so we repeated this experiment with mice that have intact CD25 but are lacking the IL-2 cytokine. But contrary to our hypothesis, fraction D and F are just fine, and B cell development is completely unperturbed in these mice. And also here in this proximity ligation analysis, we find that CD25 does actually not bind to any of those other chains of the IL-2 receptor and does not respond to IL-2. So it's not true that CD25 is in any way related to IL-2 signaling in B cells. Instead, we found in our proximity ligation assays that CD25 associates itself with the signaling chain of the B cell receptor, which again is responsible for the Goldilocks principle to keep intact an equilibrium, an intermediate realm of signaling intensity. And that's the case in resting B cells, but it's also even more so the case after the B cell receptor was stimulated with an anti-IgM antibody. This is actually ongoing work in our lab by Jae-Wong Lee, who's a research scientist in my group. What he found is actually that CD25 negatively regulates B cell activation, and in the absence of CD25, mice develop spontaneous germinal centers. So even without any immunization, these B cells are autoreactive, they escape negative selection, and they form autoreactive spontaneous germinal centers that are antigen independent. The other observation here was that if Jae-Wong deleted CD25 in human lymphoma cells, they undergo a peculiar pattern of autonomous calcium signaling. They have autonomous ERK activation. These cells are proliferating very fast, but also have a short half-life and die quickly, which is reflected here by expression of p53. So apparently these cells are just easily exhausted and lack competitive fitness. So we confirmed this here in a leukemia model, whereby we transformed CD25 mouse cells with the FLOX CD25 allele. And then after activation of CRE, we lose here CD25 expression on the surface. And then soon after, these cells disappear from culture. They fail to form any colonies. They cannot initiate leukemia. And the mice that bear those leukemias recover and survive for indefinite periods of time. Now, interestingly, and that's coming back to signaling feedback control, we found that upon deletion of CD25 in a similar way like deletion of P10 and SHIP1, we see that the balance of B cell receptor signaling strength is lost because we have hyperactivation of kinases, substrates downstream of the B cell receptor, including SYK, and then loss of phosphatase activity markers for P10 and SHIP1. So we think those phenomena might actually be related. That CD25 plays a role in maintaining the Goldilocks principle by regulating kinases and phosphatases. Now, how is this possible? So the tail of CD25 is very short here, and it's just 13 amino acids. And so we looked at what CD25 might bind to, how does it interact with the cytoplasmic tail. And as a negative control, and we're using this for a lot of different experiments, we introduced a mutation of the central serine residue, which destroys the main protein kinase C-beta consensus motif. And here we are using a bio-ID approach, which is based on fusions between the CD25 tail and BIR-A, which is the bacterial biotin ligase, which attaches biotins to proximal proteins based on the amount and the proximity. And as expected, we found that 2 phosphatases, SHIP1 and PTPN6 here, are in proximity of the tail of CD25, and this is not the case if the tail here is mutated. And this is also confirmed here in a more traditional experiment based on pull down and co-IP. So SHIP1, P10, and PTPN6 can all bind, but binding is weakened or entirely lost when this tail here is mutated. So in terms of function, this could be confirmed that indeed CD25 functioned as a powerful negative regulator of signaling strength. So if we inducibly activate wild-type CD25, we can block the calcium signal here that would otherwise be elicited. So if we express CD25 in its wild-type form, the signal is delayed and almost entirely lost. If we express the mutant, the suppression can still be seen, but it's much less compared to the wild-type form. And then in terms of leukemia, survival, and growth, we used here chimeras between the extracellular part of CD19, which is the B-cell specific transmembrane protein, and tail of CD25 either as wild type or as mutant. And the wild type form can rescue survival of leukemia cells, but the serine 268A mutant cannot. So again, showing that the ability to recruit phosphatases to negatively regulate signaling is important for survival of these leukemia cells. So we have modeled the interaction between these molecules and came up with a structural model for this, which has REC1, a PKC beta scaffold at its center, wrapped around by PKC beta. And this interaction here is facilitated by the CD25 tail, which inserts itself here. So overall, we think that B cells activate CD25 downstream of the B cell receptor via SYK PKC beta phosphorylation of CD25, which then forms a complex with REC1 to recruit phosphatases here at the surface, which then again provide negative feedback control. So it's like a circle that goes back to maintain equilibrium, Goldilocks principle again, for the survival of these cells. So our conclusion is that we think that negative selection can be leveraged indeed for potential therapeutic benefits of B-cell leukemia and lymphoma, either by inhibitors of phosphatases, hyperactivation of SYK, or interference with CD25 feedback control. So the goal here would be to push cells that are here at the upper limit. They're already transformed. They have a powerful activation signal over the edge by removing feedback control and balance, which then will trigger negative selection of what looks like at the level of signaling autoreactive B cells. Coming back to our database, which we find as a very rich resource for new ideas and concepts, we found another interesting outcome predictor that I would like to introduce to you. It's a recent publication that just came out a couple of weeks ago. And that is focused here on a molecule called IFITM3. And it's an interferon-inducible transmembrane protein. And as I showed you for CD25, this molecule is an outcome predictor in various B-cell leukemia and lymphoma subtypes. And it's known for a long time. Initially, it was found as a specifying molecules for primordial germ cells. And then more recently, it was found as an antiviral protein that can restrict viral replication. Here shown HIV, but more recent data shows it's also important for the restriction of coronavirus and many other viruses. And what was important to us is that it's actually used as a diagnostic tool for pediatric leukemia to find patients that are at high risk. So it's one probe set on a low density array to identify patients that are at high risk. So we studied the function of IFITM3 in a genetic mouse model and found actually that B cells, surprisingly, that are lacking this interferon-inducible transmembrane protein have a defect in PS3 kinase signaling, and they are prone to cell death, as shown by p53 activation and loss of BCL2. And importantly, these cells actually cannot be properly activated to undergo affinity maturation. So here is PNA is the germinal center marker, which is a surrogate for affinity maturation in B cells. So if these mice are immunized, they nicely form germinal centers. But if B cells that were adoptively transferred are lacking IFITM3, the amount of germinal centers or germinal center B cells and subsequent affinity maturation is drastically reduced. And the same is true in leukemia. So IFITM3 deficient leukemia cells cannot form colonies if transformed as BCR-ABL or NRAS. They also lack the ability to initiate leukemia in those two models. And they have a similar phenotype as I showed you in normal B cells in terms of lack of PS3 kinase signaling, survival, and stronger expression of death-related or checkpoint-related molecules like P53. So in terms of structure and mechanism, we were able to figure out how IFITM3 is regulated in B lymphocytes. It's actually a very short protein of only 133 amino acids in length, and we found that it can actually insert itself into the cell membrane. And this happens when downstream of the B cell receptor, LIN and other Src family kinases phosphorylate IFITM3 at this tyrosine 20. So that's really a central tyrosine, which leads to recruitment to the cell membrane, and then it can either interact with the B cell receptor or it becomes internalized again. So for this reason, we studied this mutation here and actually found that it can function as an oncogene. So when we introduce this phosphomimetic form of Y20, which mimics a conformation that is always phosphorylated, into a mouse strain that carries transgenic BCR-ABL, which has a very long latency to disease, we actually found that this leads to increased formation of colonies, increased PS3 kinase activity, and also increased activity of the B cell receptor signaling pathway. And so structurally, we could show also in an interactome analysis that this form of IFITM3 intersects with multiple central components of both the PS3 kinase and B cell receptor signaling pathway. And that's also shown here by these red dots in this proximity ligation assay, where IFITM3 molecules come in close proximity in these cells. Now, the structural basis for that is that, and that was very surprising to us, that IFITM3 can directly bind to PIP3, which is an initiating phospholipid and lipid rafts to initiate PS3 kinase signaling. And that is unexpected because this interaction is usually mediated by a so-called pH domain in larger proteins. But IFITM3 is such a short protein that has no resemblance of a pH domain. So we were looking here for a new structural basis of how this interaction could happen in the absence of a pH domain. And we looked at the conserved intracellular loop of IFITM3 that is used to insert into the cell membrane. And in doing so, we found a cluster of 5 basic amino acids, and of particular interest is this bracket here, we would call it, between lysine 83 and lysine 104. And even though they are 21 amino acids apart from each other, they come very close here in the structural analysis, and they basically form a clamp to directly interact here with this PIP3 molecule. So by mutation analysis, we were able to show that the wild type molecule with this bracket of lysine 83 and lysine 104 intact is a powerful initiator of PS3 kinase and B cell receptor signaling. But when these 2 amino acids here are mutated, so the bracket is no longer active, we entirely lose this ability. So that's something that we become really interested in, and that we hope to pursue further in collaboration with our colleagues at Yale, like a new way of how proteins can make contact with PIP3 to initiate PS3 kinase signaling in normal and oncogenically transformed B cells. So our model is that in the absence of IFITM3 in normal cells or also malignant cells, the molecules that initiate PS3 kinase signaling are scattered throughout the cell membrane. Only if IFITM3 is there acting as a molecular glue, they are drawn together and form a tight complex to initiate signaling. I'm now coming to the last part of my talk, which was quite surprising to some of us, and we're still looking for ways to translate that knowledge, looking for how this can be exploited therapeutically. But it essentially starts from the question of how do oncogenic pathways, once they're activated by mutations, how do they interact and become part of an orchestrated move that leads to malignant transformation. And this idea is based on a concept that was formulated and a long time ago by Fearon and Vogelstein here, the iconic concept of multi-step malignant transformation by sequential acquisition of driver oncogenes that together then form the development of colorectal cancer. So the question here is this the same in B cells? Is it true that acquisition of additional mutations lead to more malignant phenotypes, and how do these oncogenic pathways interact with each other? So to answer this question, we formed a collaboration with the Children's Oncology Group and St. Jude and studied mutation data from 1,148 cases of B-cell lineage ALL. And what we did first was what we would call a mapping analysis of affinity versus repulsion of pathways. So basically asking the question, so activating lesions in one pathway, are they, do they have affinity to activation of in a different pathway, or is there like relationships of mutual exclusivity? And we found a number of interactions that we're still working on, but one was really striking to us, and it's an interaction of repulsion, of mutual exclusivity, as is shown here in this cartoon, and that involves a STAT5 pathway, the JAK STAT5 pathway, and the ERK or MAP kinase signaling pathway. And here I'm showing you the result based on these 1,148 cases. Many of them have shown here in green STAT5 activating lesions forming one large cluster up here, and then here's another cluster, but these are ERK activating lesions. And only in 35 cases, which is just 3%, we found activation of both pathways, which is much lower than random. Also, when we look at individual cases and look at phosphorylation of ERK or phosphorylation of STAT5, we have a clear-cut negative or inverse relationship between them. And you can also see here at the level of Western blot that you have either phosphorylation of STAT5 or phospho-ERK. And this leads also to different profiles in terms of drug sensitivity. So trametinib is a MEK inhibitor within the ERK signaling pathway that affects these leukemias here, whereas ponatinib affects mainly the STAT5 signaling pathway, which is affecting those leukemias here. And so we became interested in this small minority of cases in which we have indication of activation of both pathways, even though they seem to be mutually exclusive. So we wanted to know, do they occur in the same cell, or how does this work? And to answer this question, we developed in our lab a single cell phosphoprotein analysis that allows us to interrogate phosphorylation of STAT5 and phosphorylation of ERK concurrently in single cells. And this is here based on a gel matrix where we can deposit 6,400 cells, single cells, and then look at STAT5 and ERK phosphorylation events individually. And these here are 4 patient-derived cases where we looked at individual cells and were then actually able to determine that even though for all those 4 cases we get a dual signal by Western blot, if we use our single cell phosphoprotein analysis, we see that these are actually 2 competing clones. So one clone here is STAT5, the other clone here is ERK, and that goes for all 4 cases, and we don't see any double expressing cells. So our conclusion is that these are actually rare biclonal diseases in which 2 clones are competing against each other. Then we asked, so what is the reason for that? So what is the underlying mechanism that these 2 pathways just can't go together? And to address this question, we actually foisted the alternative pathway on the leukemias that are driven by the other pathway, meaning that here is a BCR-ABL or STAT5-driven leukemia, then with NRAS, or an NRAS-driven leukemia with BCR-ABL, and we use different models for that. And here, this colony-forming assay shows if you have one single driver, either in the RAS or in the STAT5 pathway, this dramatically increases the number of colonies, but if we have both together, we basically lose all colony formation capability. And the same holds true for growth. So single driver nicely lead to outgrowth, but combination of both is suppressive. What was really surprising to us is actually that if you use genetic ablation of the diverging or alternative pathway, even though we basically remove an oncogenic driver, this actually leads to acceleration of leukemia initiation in this model. So in this case, we have here a BCR-ABL or STAT5 driven leukemia, and we remove ERK. This accelerates development of leukemia. Likewise, in a KRAS driven leukemia, removal of STAT5 initiates faster development of leukemogenesis. And biochemically, we were able to recapitulate this with small molecule inhibitors, so trametinib, a MEK inhibitor, extinguishes ERK kinase activity as expected, but it also induces phosphorylation of STAT5, and ruxolitinib here has the opposite effect. It distinguishes STAT5, but increases ERK activity. And so to end here this final chapter of my talk, we came across agonists that we use for pharmacological reactivation of diverging pathways. So in this case, BCI here, BCI 215 is a powerful activator, an ERK agonist, strongly activates phospho-ERK at the expense of STAT5, and then DPH here is a STAT5 agonist that drives STAT5 phosphorylation, but at the expense of ERK. And this has interesting activity on the clonal dynamics of these leukemias in vivo. So STAT5 leukemia can be converted into an ERK leukemia. And most importantly, if you combine these pathway agonists with conventional treatment, so for instance here an ERK inhibitor with a STAT5 agonist, we achieve a dramatic prolongation of overall survival. So the final conclusion here is that we propose that diversity of signaling input is actually an important barrier of malignant transformation, and centralization and convergence onto one single pathway, inactivation of all the other pathways, is an early and critical step of malignant transformation. And if we achieve to reinstate a diverse signaling environment that would resemble the interactions of normal cells with the environment, which have multiple receptors, multiple cues from the environment. So we are proposing a strategy of pharmacological reactivation that would restore the diverse signaling environment. And we hope that this approach can also be leveraged to overcome conventional mechanisms of drug resistance. So here we have pathway convergence, minimal what we call friction, and a permissive environment for transformation. But here, if we have divergent pathways, we actually do have some friction and create a non-permissive environment. And with that, I would like to thank a number of collaborators at Dana-Farber, at Yale, and would like to acknowledge in particular two senior members of my lab, Linda Chan and Jae-Wong Lee, who did most of the conceptual innovation of this work. Thank you very much.
Markus, that's an amazing collection of studies, and it is remarkable how complex and somewhat almost counterintuitive a lot of these pathways are in B cells and B cell malignancies. And it's obviously a delicate balance. And let me ask you, because you've identified a number of pathways that I guess are principally designed for B cell elimination, that you could leverage. How would you potentially target those pathways, or could you target those pathways in conjunction with the growing armamentarium of available therapies for B cell malignancies that are now in practice? Or could you leverage that combination?
Right. In terms of potential for translation and how we would leverage hyperactivation of B cell receptor signaling to engage negative selection, I think we have two options. One is already available but less attractive. That would be inhibition of phosphatases like SHIP1 and PTEN. Unattractive because in the field phosphatase inhibition raises a lot of questions about specificity. An alternative approach is one that Mark Lemon and I have discussed, and that would be to generate a direct agonist of a key kinase, like SYK or BTK, something fairly upstream in the B-cell receptor signaling cascade. So actually yesterday, I was at Mark's CBI weekly meeting, and there was some encouraging feedback that that might actually be feasible. So that's an approach that I would definitely like to pursue in the future, to develop a direct hyperagonist of one of these key kinases.
Excellent, thank you. So, and people should feel free to submit questions on the chat box. Diane Krause has a question. I don't know if you can see it, Marcus. I'll just read it. PLA suggested that CD25, CD79A colocalization is predominantly intracellular. What do you think that this indicates?
Great question. So we have done an experiment with WGA, which is a surface marker, and we actually find colocalization of about 60 to 70% of those interactions with CD79A and B. So I don't think it's predominantly, but the question remains, there's still a significant amount of interactions that are within the cell. And we thought, indeed, that is very strange, because why would the B-cell receptor be internalized or somewhat within the cell associate with CD25? And so we can't answer this fully, but in light of a recent study by Lou Stout that was published in Nature 2 years ago, where he actually shows that in malignant lymphoma, B-cell receptor signaling complexes form off of endosomal membranes within the cell. We think that the exact same thing might happen here. We don't know that, but that would be our explanation. So I don't think it's predominant, but I agree with Diane that we see intracellular complexes and we think they are in endosomes.
Thank you. And Diane has a second question, which is CD25 seems to prevent autoreactivity. Do you think that this is related to CD25, CD79A interaction? Does CD25 interact with surface immunoglobulins?
So that's actually a question that I asked Eric Meffre, who's my collaborator in the field of autoimmune diseases. So Eric told us that CD25, and that's known by work from his group and also others, is crucial to maintain central B-cell tolerance. So if that molecule is not there anymore, then central tolerance mechanisms don't work. I think our signaling study is just about to clarify how this actually, what is the mechanism of that. I think the link or how CD25 interferes with B-cell receptor signaling is not known, and our paper is not published yet, so we're still working on that. And I do think actually that it does interact with surface immunoglobulin, yeah.
And again, people should submit their questions online. Marcus, I mean, could you ever conceive of, you identify a B-cell malignancy that's driven by JAK-STAT and you would give them an ERK activator? I obviously we focus on how to inhibit the pathway in cancer, but is that something you could conceive as a therapeutic approach?
If I may go back on my slides, because there's one that I want to show you. There's a study. Yeah, this study here, oops. By Veronica Sexel's group. So that's
did you want to show your slide, or?
Yes, I'm going to show the slide again. Okay. So I hope I got the question correct. This is what I would like to refer. It's a study by Veronica Sexel's group, and that it's a trial for patients with NPN, and they receive ruxolitinib, which is a JAK-STAT5 inhibitor, over long periods of time. It's actually going in opposite direction. So they found that these patients developed in 6%, so 9 patients out of 157 with NPN developed a high-grade B-cell lymphoma that were driven with KRAS. and that's a 15-fold increased risk. So what they said in the study is that actually, for unknown reason, ablation of the STAT5 pathway enables the transformation of a pre-malignant B-cell tumor that carries a KRAS lesion, and that's essentially what we did in our genetic experiment. So I think it can cut both ways. So it can be beneficial if we find ways to leverage this activity to completely suppress oncogenic signaling. But like Veronica's MPN study shows, it can also go in the opposite direction. If you try to achieve long-term suppression of one pathway, we might inadvertently activate the other.
So it's really interesting because obviously, if you're going to use ruxolitinib, its context may be very specific. I mean, I know it's a small proportion, but that's a pretty high risk. Just wait, see if any other questions. Well, I think we're, really at the top of the hour. So, want to thank Markus for really an extraordinary talk that's creating so much insight into the biology of B cells, both with respect to cancer and autoimmunity. I want to thank Dr. Snyder, Ed Snyder, for sort of continuing to lead this lectureship. And I want to thank the Frisbees for their continued support of our Cancer Center and the mission and the support of this lecture. I wish you all a great day and Happy New Year. Thank you.
Thank you very much.
Thank you very much.
Thank you, Marcus. Take care.